Hi, this is Dr. Goldsmith again. This is the second of the various recordings that I'll be putting online for you in my absence. So we have an idea of how we get recombined gene segments for our T cell receptor and B cell receptors. With that knowledge, we can now understand lymphocyte development, and that's what I would like to continue describing herein. There are some basic principles for lymphocyte development, and that is that the essential properties for mature cell function are gradually acquired. Each step is going to be progressively more limited. So as you move on, you'll move away from the pluripotent stem cells in the bone marrow to becoming a more differentiated type of cell, either a B cell or a T cell. At each step, you lose the properties of the immature cell. And once you have lymphocytes, they're committed to becoming lymphocytes. They're committed to the lymphoid lineage, and then they're either committed to the B cell or the T cell lineages. And of course, the development requires signals, and those signals are provided by the cell's environment wherever they are, which is very, very nice. So let's talk specifically about B cells first, and then we'll talk about the T cells. So the initial environment and the final environment for the B cell is the bone marrow. And B cells are going to be defined by various steps in the assembly and expression of their functional antigen receptor genes, which is why I just described that, and by distinguishing features for each type of cell. Each step along the way is going to be monitored as the B cells are developed, and each successful gene rearrangement is going to result in a protein chain that serves as a signal for the next step, and you'll see that in just a moment. It means that there are going to be specific checkpoints that occur throughout the development of the B cell, so that the B cell is going to express only one receptor specificity. And I will say that there is a lot of loss. You know, if you have random B cell receptor gene segments, randomly getting together, they're not all going to be useful. So you have to check along the way to make sure you have a useful receptor. The stromal cells, that is the connective tissue cells within the bone marrow, provide that nice environment, as I mentioned earlier. They are going to produce external signals that switch on key genes for the development. And they're going to form specific adhesive contacts with the developing lymphocytes via cell adhesion molecules. I'm not going to name them all, which I'm sure you'll be very happy about. They also provide soluble and membrane-bound cytokines and chemokines that control the differentiation and proliferation of the B cells in this case. This shouldn't come as any surprise to you. These are things we've been talking about all along, that cytokines are going to provide particular signals, and we're going to have cell adhesion to keep cells in locations for the period of time that they need to be there. So the development of the B cell occurs in stages, from the pluripotent stem cell to a pro-B cell to a pre-B cell to an immature B cell to a mature B cell. And I have this shown in an image in just a moment. The stages are going to be marked by different cell receptor markers, by the cell surface markers, by the receptors themselves, in this case a B cell receptor, and by transcription factors. I'll mention some of these, but not all of them. The stages are going to be marked by rearrangement of the immunoglobulin genes, that's for the B cell receptor. And for the first step, from that stem cell to the pro-B cell, it means you're committed to the B cell lineage, and there's going to be lots of proliferation, which I noted in the previous recording. So lots of proliferation going on. They're committed to becoming B cells. They're not going to become a T cell. You won't get anything else but a B cell, although you may not get a fully functional B cell either. You've got to check to make sure that the gene rearrangement and production of the B cell went appropriately, and there will be checkpoints along the way. The next step is to move from a pro-B cell to a pre-B cell. Again, there's going to be a lot of changes. The pro-B cell will enlarge to become a larger pre-B cell, and it involves lots of cell division. The population is going to expand 30 to 60 times, so that's a lot of cell division. And during this process, there will be successful VDJ rearrangement of one heavy chain locus. So, one allele from one parent, the gene loci will rearrange as was just described in the previous recording. You'll get one mu, always the first, heavy chain protein. Most of that is actually going to stay within the cytoplasm of this developing B cell. Some of the mu protein is going to be expressed on the surface of the cell in association with two other invariant surrogate, meaning they're not the actual light chains, they're stand-ins for them. So that mu protein that underwent rearrangement is going to be expressed on the cell surface with two other invariant light chain proteins, and it's going to be checked to see, is this a good B cell receptor? In other words, the mu heavy chain is going to be taken out for a test drive along with the surrogate light chains. There will be Ig alpha and Ig beta signaling molecules, and all of that together is going to form a pre-B cell receptor complex to check, does this B cell receptor complex pass muster? If it does, assuming that there was successful expression of that mu protein and the B cell light chain invariant chains, the B cell receptor complex, the pre-B cell receptor complex, two other processes will be signaled. The first is allelic exclusion. This is a process by which recombination of Ig heavy chain gene segments on the second chromosome from the other parent is going to be shut off. So you only get one allele expressed in each developing B cell. The second process that is going to occur, assuming there was this successful test drive of that pre-B cell receptor, the second process is recombination of immunoglobulin light chains. The first recombination event will involve kappa light chain gene segments. If that is functional, it associates with the mu chain that was already successfully developed and will form a complete membrane-bound IgM receptor. If the kappa chain recombination doesn't go so well, lambda chains will try. And then again, the cell will check to see if it is appropriately expressed. So the light chain rearrangement is going to occur through V and J gene segments being rearranged. Remember, they don't have D segments. It's going to occur one allele at a time, so allelic exclusion is going to apply here too. And this rearrangement can occur several times if the first attempts don't produce a fully functional productive rearrangements. Assuming you do have a fully functional completed IgM antigen receptor, so you have appropriate IgM antigen receptors expressed, then you get a shutoff of production of the recombination enzymes, so the RAG enzymes and the other enzymes that are associated with those recombination events, and further recombination of any unrecombined light chain loci that ends. It's shut off. So B cells are going to express either one kappa or one lambda light chain, and and this is going to be inherited from one of the parental alleles, not from both. So having the kappa and the lambda, possibilities, it just increases the chance of having a successful gene recombination and expression of a functional receptor. So remember, there's a lot of recombination. It's totally random, and you're going to lose a lot of B cells, but with the increased chance, some of them will actually pass muster. So once you have this membrane-associated IgM receptor, this is a second checkpoint. You check to see whether that is appropriately expressed. If it is, this is now an immature B cell. And now you have to look to see, is this B cell going to bind host antigen with too high an affinity? I sort of liken this to a Goldilocks principle. If it's too strong binding self-antigen, that is negative selection. Negative selection is going to occur. If self-antigen binds with a really high affinity, further maturation is stopped. It is possible to get VDJ recombinase started again and have receptor editing occur. This occurs in B cells, but not T cells. And you might get further rearrangement that would be functional. But again, if that rearrangement occurs and it binds self-antigen with too high an affinity, that cell is no good and it will die via apoptosis. If the binding of the antigen receptor to self-antigen is okay, so not too great, then it will go through the process of final maturation. And that final maturation involves co-expression of both the IgM receptor and the IgD receptor. This simply happens through alternative splicing. So the recombined VDJ heavy chain RNA may be spliced onto the mu RNA or the delta RNA. This is alternative splicing, and so you get either a mu or a delta RNA and expression of IgM receptor or IgD receptor. These both being expressed on the B cell developing, so if you have an IgM positive and an IgD positive cell, both of them are expressed. That is a mature but naive B cell. Again, we've got to go through the process of selecting for cells that are bind okay, but not too much antigen, and they need these checkpoints to make sure that we've got binding that's okay. If it's too strong, then apoptosis will occur. Negative selection only for the B cells. There's no positive selection. That only occurs in the T cells. So here's that process illustrated. You have your your Ig, DNA, and RNA, and whether there is expression of the protein from the stem cell where you have germline DNA being expressed to a pro-B cell, it's still germline DNA, to a pre-B cell where you've got a recombination of the heavy chain first, so VDJ recombination associated with the mu constant region. That is going to be expressed on the plasma membrane as the pre-B cell receptor. One heavy chain that worked in combination with the surrogate light chains. And a lot of the mu B cell receptor protein will actually remain cytoplasmic. If it passes this checkpoint, so this is one checkpoint, it will continue its maturation process, recombining of the light chains, kappa first, then lambda. And then if that works appropriately, you would get expression of a fully formed IgM receptor. So here it is being expressed on the immature B cell. This is another important checkpoint. If it passes this, you get further maturation to the mature B cell where you get alternative splicing, so you get both the mu and the delta RNA, so you can express both membrane IgM and IgD will be expressed on that mature but naive B cell, meaning it's never seen microbial antigen before. So that's the process for the B cells. What about the T cells? Very, very similar to what happens with the B cells. The development occurs in a stepwise manner. Each step is going to be monitored. It includes rearrangement of those antigen receptor genes, and there are going to be checkpoints throughout. So that ultimately a T cell expresses a successful receptor that's a complete, in this case, heterodimer, because the T cell receptor is an alpha and beta, the vast majority. There are some other processes, though, because although I say, the alpha-beta T-cell receptor is the vast majority of the T cells, there are some gamma-delta, although I'm not going to focus on those. So T-cell development, there are some twists. So again, there is going to be a lymphoid progenitor in the bone marrow. So T cells all start off in the bone marrow to develop, but then some of these are going to travel to the thymus, where the rest of the development is going to occur. So the cells that move on are going to pass through a series of distinct phases, and there are going to be changes in the status of the T cell receptor genes, the expression of the proteins of the T cell receptor at the cell surface, expression of other cell surface proteins like CD3 and zeta. And remember that T cells have co-receptors, that is CD4 and CD8. So when you talk about T cells, when they arrive at the thymus, they're known as thymocytes, and they actually don't express CD4 or CD8. They express neither. They are considered double negative thymocytes. Double negative meaning neither CD4 nor CD8. At some points, both CD4 and CD8 will be expressed, and these are known as double positive thymocytes, and I'll tell you when this occurs. And then ultimately, you're only going to have one co-receptor express either CD4 or CD8, and those are known as single positive thymocytes. So what exactly is going to happen? Well, there are stages, again, just like with the B cell. The T cell moves from a stem cell to a double negative T cell, also known as the pro-T cell, to a pre-T cell, to a double positive T cell, also known as an immature T cell, to a single positive T cell. Like the B cells, the stages are marked by cell surface markers, by the receptors that are being expressed, and by transcription factors. And a key among this are checking to make sure that there is appropriate rearrangement of the T cell receptor genes. So, the cells that arrive at the thymus are going to be double negative pro-T cells. They arrive there, and if they undergo appropriate rearrangement of the beta chain of the T cell receptor, again equivalent to the heavy chain of the IG receptor, the first rearrangement will be the beta chain, so VDJ rearrangement, to produce what is known as a pre-T cell receptor. Again, this is occurring in the thymus. This will be beta chains that have been rearranged, their gene segments, and a surrogate alpha chain. Again, very similar to what you see with the B cell having the IG heavy chain and a surrogate light chain. This is known as the pre-T alpha receptor, and it's expressed with CD3 at the double negative stage, so neither CD4 nor CD8. In other words, this is really analogous to the pre-B cell receptor, and it's going to be a key checkpoint. Again, the thymus is going to provide the appropriate environment from the stromal cells, like that IL-7 that was mentioned for. Lots of proliferation is going to go on, but a lot of these cells are going to die because they don't have the appropriate rearrangement of their beta chain and expression of that with the surrogate alpha chain. So you need lots and lots of proliferation. If there is appropriate expression, these pre-T cells can move on. The expression of these pre-T cell receptor complex with CD3 causes lots of cell proliferation and enlargement. Arrest of the beta chain rearrangement, so allelic exclusion is going to occur. Again, very similar to the B cell receptor rearrangement. And then you get expression of both CD4 and CD8. In other words, the cells are going to become double positive. Proliferation is going to cease, and when this happens, the cells are considered small double positive thymocytes, and the alpha chain rearrangement will occur. So these are now immature T cells. They may express an appropriate alpha chain if the rearrangement goes appropriately. They will express this at low levels. This is going to be another key checkpoint. So this will be now a fully functional T cell receptor. It's not a surrogate anymore. The beta chain already passed muster with a surrogate, and now we had the alpha chain rearrangement occurring. And at this point, the T cells undergo both positive and negative selection. And again, not all of the cells will pass this test. Positive selection means that only the T cells that are capable of recognizing self-MHC are going to be selected. So any of the thymocytes with T cell receptors that interact with self-peptide and self-MHC complexes are going to survive and mature. But lots of them are going to die via apoptosis, and this is going to occur in the thymus. So you get MHC restriction. Again, it's like Goldilocks. Not too much, not too little. So, recognize MHC plus antigen, that's good. It's only for the T cells. The B cells don't need this, so positive selection only occurs in the T cells. And ultimately, the cell, as it's maturing, will express either CD4 or CD8. I will go over how that will happen in just a moment. It really depends on the specificity of the T cell receptor for self-peptide, self-MHC molecule. So I'll talk about that when we get to the images. Negative selection is also going to occur at this point. This is going to get rid of any T cells that react too strongly, just like the B cells reacting too strongly. This is negative selection. So too strongly with self-MHC plus self-peptide complexes, the T cells are going to get eliminated. In other words, you want them just right binding to self-MHC plus antigen, and that is going to ensure that we're tolerant to oneself. Most of the cells, again, are going to die. They'll die via apoptosis, so that's why a lot of proliferation was required along the way. So here we have what was just described in text. This is a picture just like was shown for the B cells. We have our stem cell in the bone marrow. Some of these are going to be of the T cell lineage. They will move to the thymus and arrive as double negative pro T cells. No CD4, no CD8. Double negative thymocytes. The first thing that happens is rearrangement of the beta chain locus. So you get a beta chain, gene segment rearrangement, and a surrogate alpha chain. So you get the pre-T cell receptor. So a surrogate alpha chain and the beta chain that has been rearranged. If this responds appropriately to the environment, then the cell is allowed to continue on. This pre-T cell with its pre-T cell receptor will then start expressing CD4 and CD8. The alpha chain will undergo rearrangement, and you will get expression of a full T cell receptor. That is not enough, though, because you have got to select appropriately. We have positive selection that goes on. If you have a double positive immature T cell that happens to recognize MHC class II plus antigen, and it does so appropriately, you're selecting positively, and it will be a CD4 T cell because the co-receptor recognizes MHC class II. Again, it's got to be just right, not too strong, not too weak. If you have a just right association of the rearranged complete T cell receptor and it recognizes MHC class I, then you will have a mature CD8 T cell. So you're selecting for recognition of MHC plus peptide. And again, these are host peptides, not too strong, not too weak. If there's no recognition, so co-receptors don't recognize the MHC molecule and the T cell receptor is not working appropriately, then the cells will die via apoptosis. In other words, they fail positive selection. And that occurs only in the T cells, but in a similar way to the B cells, negative selection will also occur. If the recognition of MHC plus antigen is too strong, that's no good either, because you have the possibility of harming yourself, recognizing self-antigen later on if it's too strong. And those would be negatively selected, meaning they would also die via apoptosis because they recognized MHC plus antigen too strongly. So the twist here is you've got to think about the co-receptors, CD4 and CD8. If it recognizes MHC class II, it's going to be a CD4 positive T cell, assuming that the T cell receptor is okay. If it recognizes MHC class I plus the antigen, it will be a CD8 positive T cell, single positive ultimately, as long as the T cell receptor is okay. And again, too strong recognition either way. It's negatively selected, meaning it will be taken out via apoptosis. So essentially, lymphocyte maturation and survival is really regulated through the antigen receptor. Strong signals to an immature lymphocyte, the cell dies or further receptor rearrangement can occur. And the idea here is if it's too strong, it's going to delete any self-reactive receptors. This is that concept of negative selection. If there's a complete absence of signal, it's also going to lead to cell death. That's no good either. The receptor is probably not useful. It doesn't recognize anything, so dies via apoptosis. Periodic signaling, it's just right. This is the idea of the Goldilocks principle. Okay, we've got a B or a T cell receptor. It's just right. And so self-reactive cells and cells with no signal are going to die via apoptosis. So what are the signals? It really seems like they're delivered by the lymphoid organs, at least partially from the self-antigens, but we don't know all of them. But certainly the environment matters. We do know that. All right, so given that, I would like you to answer the following question. Think about it, and I'll show you the answer after you've thought about it. Which of the following is false? Only T cells undergo positive selection. All immature B and T cells will become mature cells. Both T cells and B cells must pass checkpoints during maturation. Both T cells and B cells undergo negative selection. And there is only one correct answer here, and it is all immature B cells and T cells will become mature cells. That's not true. There is a lot of loss along the way, lots of death via apoptosis because they don't express an appropriate antigen receptor. They recognize self too strongly or not at all. In the case of the T cell, they don't recognize MHC. They have to recognize MHC. That's positive selection. And in the case of both the T cells and the B cells, they have to undergo negative selection. That is true. So 1 is true, only T cells undergo positive selection. 3 is true, they have to pass checkpoints during maturation. And 4 is true, that both B cells and T cells undergo negative selection. Not all of them are going to end up mature cells, so this is the one that is false. And that is all for this segment. Thank you for listening.